You just might bomb your preparations for that exam. And perhaps that's because your attention is distracted away from the studying, or your emotions are interfering with your performance. But many studies have been conducted on the effects of stress on the neurobiology of learning and memory. And frankly, the overall picture is a bit of a mess. Many labs use combinations of stressors that might be as artificial as foot shock or as natural as exposure to the cold. But even these kinds of manipulations can be very problematic. I'd like to define briefly that we should consider stressors falling into at least two distinct categories, known as systemic and processive stress. Systemic stressors are physical stressors that convey information about the status of the body to the brain. This includes information about the nutritional and thermal status of the body. And these stressors are mediated by brainstem inputs to the hypothalamus so that the animal can mount an appropriate hormonal and physiological response, like increasing circulating concentrations of cortisol or corticosterone to combat the imbalance. Personally, I feel that this type of systemic or physiological stressor just is not as interesting for psychologists because this kind of stressor doesn't really portend alterations in limbic function. And this kind of stressor doesn't predict pathology. For example, marathon runners stress their bodies immensely. And every time they go out for that run, they have huge increases in circulating cortisol. But they're not particularly vulnerable for major depressive disorder. The other category, processive stress, refers to things that you must identify as impending challenges to homeostasis. These are stressors that activate the limbic system to trigger increases in arousal, alterations in cortisol responses again, and changes in affect and behavior. These are the stressors that we as psychologists are more interested in. The main challenges that these stressors represent can be summed up as a loss of control over the environment or lack of predictability in the environment. And if you think about it, those are the factors that define stress for you too. Loss of control over the environment and lack of predictability in the environment. So your wife dies, you crash the car, you lose your job, and the kids are on drugs. These are all processive stressors, all processed by the limbic system, and all predispose vulnerability for various alterations from normal, healthy brain function. So these are the stressors that I think are more interesting for studies of the neurobiology of learning and memory, especially in relation to the severe impact of a lifetime of processive stress as we age. There's now an abundance of processive stress throughout the life. There's an abundance of evidence that processive stress throughout the lifespan damages the hippocampus and is an important contributing factor for age-associated memory impairment, or AAMI. Okay, the photo that you see at the left, also from my lab, is a stress paradigm that I advocate and that we've used quite extensively. It's called social defeat. In this paradigm, rather than exposing the rats to a cold room or a food deprivation or a foot shock, what we do is we have a large, sexually experienced, and therefore territorial dominant male rat. And that rat is housed alone in a standard shoebox cage, as illustrated in the photograph. That big male resident rat is the one on the left. And then a smaller, young, and inexperienced male rat is placed into the cage. The dominant male rat will investigate the intruder and then pin it down, standing over it for a few seconds. This is dominance behavior. The intruder will display submissive behavior and mount a massive corticosterone response. And the pair will engage in this interaction repeatedly until we actually remove the intruder from the cage, and perhaps test it again with a different dominant male the next day. We think that this interaction models the kinds of social stress that we may experience in our daily lives in occupational and familial and romantic and other kinds of relationships. It's a much more natural model than artificial foot shocks, and much more emotionally salient than cold rooms or food deprivation. And it doesn't confound the difference between systemic and processive stress. So I'd actually like to see a lot more studies of social defeat stress applied to the neurobiology of learning and memory, so we can develop a greater understanding of the role that social stress plays in our lives, and perhaps even contributes to age-associated memory impairment as we age. Okay, I got on a bit of a roll there. I do like to consider carefully the relevance of our manipulations for our understanding of the factors that impact us throughout our lifespan and that have the potential to alter brain function. So stress is clearly a big one, and the environments that we live in, I believe, can have a profound impact upon our learning and memory capacities as we age. But I'm going to move on to some more physical interventions here, some more intrusive kinds of techniques. I'm going to fairly quickly go through some of the ways in which stereotaxic surgery is used to manipulate brain function of rodents for studies of the neurobiology of learning and memory. At the upper left of the images on the right side of the slide, you can see a stereotaxic apparatus. This apparatus holds the anesthetized rat's head in place, and micromanipulators here and here are used to move electrodes or injectors into the animal's brain. The scalp is opened and readings are taken of markers on the skull known as bregma and lambda. And these, of course, are shown over here in the upper right with a diagram of a skull and an actual photograph of an exposed skull of a rat that's going to have surgery done on it. So once those measurements are taken, a hole is drilled into the skull at the relevant location, and using the micromanipulators, measurements are taken, and the electrode or injector cannula can be lowered into the brain to find the target location with a high degree of precision. So using an injector, the brain can be lesioned or damaged in a variety of ways to destroy an area, or chemically select certain types of neurons in an area under study. And so this is illustrated in the image at the bottom of the slide here, where you can see a target brain site right in there and there, where you might implant an electrode or a guide cannula for injections. It actually looks like the bed nucleus of stria terminalis, or BDNST, that I talked about on the previous slide. So what a lucky coincidence, right? So of course you can also use the stereotaxic apparatus to implant one or more electrodes to stimulate and record from neurons in the living animal, and this could include manipulations for studies of LTP or LTD. And you could also set up an animal so it engages actually in intracranial self-administration. This photograph is taken from an animal in a study that I conducted as part of my doctoral dissertation. So they could do intracranial self-stimulation with an electrode, or actually intracranial self-administration of a drug through a cannula. Or you could simply set up an indwelling cannula to give repeated access to the brain, so you can inject drugs either into the fluid-filled ventricles of the brain, those are known as intracerebroventricular injections, or directly into the brain region you want to study, and those are known as intraparenchymal injections. And there are also ways that you can impact brain function by systemically injecting drugs or having the rats self-administer drugs intravenously. Lots of drugs have been used and lots of experiments have been done to examine the properties of the drug formulations themselves, or to use the drugs to examine the functioning of receptors, transporters, and other mechanisms in the brain. It's probably also worthwhile to note that drugs can be administered acutely or repeatedly, or in order perhaps to avoid the stress of repeated injection or provide a more ongoing administration, an osmotic pump can be implanted, as we saw in the experiment where APV was chronically administered throughout the training in the water maze. These pumps supply a slow, continuous release of the drug by taking advantage of osmotic gradients. And so we have lots of drugs that have been used in experiments on the neurobiology of learning and memory. At the bottom left of this slide, you can see a variety of drugs that act by binding at different sites on the NMDA receptor, including the antagonists APV and CPP and MK801 and memantine and amantadine and PCP, all antagonists at the NMDA receptor. And of course over here, the agonists, glutamate, NMDA, glycine, and serine. All examples of drugs that could impact the functioning of the NMDA receptor. And we can administer them through all of these various routes that I've been describing on this slide. And then, in addition to pharmacological manipulations, we now have state-of-the-science genetic manipulations. This includes the production of transgenic animals in which a gene is either knocked out or overexpressed or modified to do something somewhat different from the original gene. And the genetic manipulations also include infection with viral vectors that carry gene constructs that can augment or alter gene function by actually inserting a gene into the cells. And then there are optogenetic manipulations that can alter the functioning of target neurons by using viral vectors to express light-sensitive proteins that can control ion channels or metabotropic actions in specifically targeted cells. Now, this section is not meant to make any student an expert in recombinant DNA technology. But let's take a quick look at these manipulations. And hopefully by the end of this, you can imagine how some of these manipulations may be meaningful for studies of the neurobiology of learning and memory. Now, the method for generating transgenic mice is illustrated in the center to the right half of this slide. Mice are bred and the blastocyst or ball of undifferentiated cells is extracted from the impregnated female. A gene construct is engineered with the manipulation of the target gene. And if constructing a knockout, the gene is altered in a way to introduce a nonsense mutation, a stop codon inserted into the gene, or a shift in the open reading frame to make a useless protein. The gene construct is inserted into a bacterial plasmid, or a kind of a circular gene sequence, and then incorporated into stem cells extracted from the blastocyst. The stem cells expressing the gene construct are injected into a blastocyst from another female, where they basically then kind of intermingle with the stem cells or undifferentiated cells of that blastocyst. The blastocyst is reimplanted into a pseudo-pregnant female, and note that this makes what we call a chimeric mouse offspring that actually has two genomes. The mutant genome containing the new construct and the genome from the original mouse. Now, scientists even combine different mouse strains to generate these offspring with different coat colors. Because with two germ lines of cells with different coat colors, you can clearly see that you have two genomes in the chimeric mouse progeny. And then the chimeric offspring are bred with standard wild-type mice. And some of the offspring will have the manipulated gene on one chromosome in combination with the wild-type gene on the other chromosome. And these are known as heterozygotes. If we then breed these heterozygote transgenic mice, then 25% will have no copies of the manipulated gene, and 50% will have one copy of the mutated gene, and 25% will have two copies of the mutated gene. Simple Mendelian genetics. So then we can study the homozygote mutants and compare them with the homozygote wild types and with the heterozygote intermediates that have one wild type and one mutant gene, so that we can evaluate the impact of the gene manipulation on learning and memory. And this kind of technology can actually have a profound impact on the functioning of the transgenic animals, as illustrated in the photograph at the upper left. In this photograph, you can see that the mice are of the standard C57 Black 6 strain. Of course, the one on the left is a wild-type C57 Black 6 mouse. The one on the right is a C57 that has had the leptin receptor knocked out. They're designated OB mice, meaning that without this gene, they do not regulate the balance between food intake and energy expenditure efficiently. So they become immensely obese. This is one very well-known illustration of the powerful impact of these gene manipulations. And we've already seen some examples of transgenic animals that have CAM kinase 2 manipulations. And so what I've just described is the technology that brings them and many other genetic mutants into being. And now we even have conditional and tissue-specific transgenic animals in which the expression of the mutant gene is placed under control of a specific promoter that causes it to only be expressed either at particular times or in particular parts of the brain, and so on. I feel like I got to rein in my commentary here because I could keep talking on and on about these technical advances, these very exciting new kinds of scientific experiments we can conduct. But anyway, that would take us on a sidetrack that we shouldn't go down too far, or we'll never cover the core material that I want to teach you. So let's do this. If anyone wants to talk about these innovations, let's do so in office hours, okay? But I do want to quickly talk about the last two manipulations. One of them is viral vectors. This is another huge advance. And this is a manipulation in which we take a vector like the one pictured right about there. We take this virus, typically an adeno-associated virus these days, and we insert a gene construct into these viruses. You see, viruses actually function by attaching to receptors on cells. They then insert their DNA or RNA into the cell and use that cell's machinery to make copies of themselves. The cells then fill up with fully formed new viruses and lyse, or burst open, spilling out all the virus particles to infect other cells. So if we were to actually strip out the pathogenic genes that encode new virus particles and insert gene constructs that the virus will then insert into target cells, the infected cells will make the proteins encoded by the gene construct that was inserted, but will not make new virus particles because we stripped out those pathogenicity genes. So the infection is limited to the number of virus particles that were injected at the target site. And we have cells now that express the genes that we made under control of whatever promoter we chose to engineer. So this is really powerful technology. It's also the basis for the concept of gene therapy. And it's used in research applications to study many issues in modern neuroscience, including, of course, studies of the role of genes in the neurobiology of learning and memory. Think about what I talked about previously, with genomic cascades being signaled. What if you could make an animal that had alterations in that genomic cascade? Or what if you made an animal that had an alteration in one of those genes that is constitutively expressed, that you have the mRNA available all the time? Modify those and see what happens in terms of the neurobiology of learning and memory to discover what's the actual role of the gene products in the plasticity. Okay, and one more cutting edge technology that I'd like to address, and that is Karl Dieseroth's optogenetics. This technology takes advantage of the fact that some genes of some simple marine organisms encode proteins that respond to wavelengths of light. So it's illustrated in the picture at the lower left, where a light-emitting diode implanted into the brain of the rat can be turned on and off at will to control the firing of specific neurons and hence the actual behavior of the rat. So let's look briefly at how that works on the next slide. The graphic at the far left shows that there are three ways that neurons can be controlled with optogenetics. In the top example right here, a gene construct is expressed encoding a channel rhodopsin that opens a cation channel to excite the neurons whenever there's blue light stimulation. The second diagram shows a halorhodopsin that actually opens a chloride channel when a yellow light is shone to inhibit the cell expressing this protein. And the bottom diagram shows an OptoXR, like OptoA1 for example, which controls a metabotropic receptor when green light is shone. So in order to accomplish these ends, the first step is to synthesize a gene construct that attaches a promoter that will target the construct to a specific type of cell to a gene that encodes a light-sensitive channel or metabotropic receptor. For example, CAM kinase 2 is only expressed in forebrain neurons, where Thy1 is only expressed in projecting neurons and not interneurons of the cortex. So you could use the appropriate promoter to drive expression of your construct only in the type of cells that you want to study. Use the promoter for CAM kinase 2 if you want to activate forebrain neurons by, let's say, turning on a blue light, or to inhibit them by turning on a yellow light. Or use a Thi1 promoter to control cortical projecting neurons. Then you package your construct into a viral vector and inject it into the region of the brain using a stereotaxic apparatus in the way I described previously. You can then control the activity of the neurons and monitor the electrophysiological or behavioral responses of the animals. The advantages of this technology include direct temporal control of the activity of neurons. You simply need to turn the light switch on or off, and you can see the consequences immediately. The advantages also include that you can control very specific sets of neurons rather than broad fields of neurons that you would impact if you just used, a stimulating electrode. And of course, you can activate and inactivate the neurons that interest you without all the messy off-target and side effects of drugs. So I hope this sounds exciting to you. It has potential for lots of uses in neuroscience studies, as well as a potential for treatment of disorders like Parkinson's disease, substituting perhaps for deep brain stimulation, and getting a much better control of only the neurons you want to stimulate, and perhaps thereby avoiding the off-target side effects of deep brain stimulation that are seen in many patients. So I hope you find these ideas for old and new ways to examine brain function stimulating, if you will pardon the pun. But there's so much more to do and so many exciting experiments to perform. We've come a long way since Carl Lashley performed knife-cut lesions all over the cortex of the rat brain to try to figure out where memories were encoded. And of course, he failed miserably in that task. Or since Donald Hebb took the rats home to play with his children and tested them to see if the enrichment improved their ability to learn in a maze. I could go on and talk at greater length about these technologies and techniques, but that, I guess, would be a different course. So I promised that this chapter would be brief, and it's gone a little bit longer than I anticipated, but I'm going to cut it off here. And hopefully that'll free up a little bit more time this week to look at chapter 11, which is a larger chapter with more to cover. But in the meantime, I'm going to return tomorrow with chapter 10.